In this lecture, we will learn about how histological slides are prepared from soft tissues. In the preparation of tissues for light microscopy, there are several processes. First is to process the tissues. Then we need to embed the tissues. Then we slice the tissues into thin sections, which we call microtome. Then we stain the tissues, and then finally we mount these tissues on glass slides. So if you take a liver tissue, which looks like this, you cut a small piece out of it and you process it, stain it, mount it, and look at it through the light microscope. This is what you will see. So you will learn in brief how a tissue of any sort, soft tissue, can be converted into a histological slide. This table shows you the different processes which need to occur for you to turn a wet soft tissue into a histological slide. Step 1 is fixation. Fixation is where we need to prevent the tissues from degrading. Once a tissue is taken out of the body, those tissues will slowly decay. So in order to preserve the tissues, we do what is called fixation. For this, we usually use neutral, 10% neutral buffered formalin. That is, you use formalin as the fixator. In general, when we take out a piece of tissue, we place it in 10% formalin for about 2 days. Then comes dehydration. That is the second step, where we need to remove the water from the tissues. A lot of water does not help us to make a proper slide. In order to remove this water, we use alcohol. Then step 3 is what we call clearing. In this, we have to infiltrate xylene into the tissues. Then the other step is to infiltrate paraffin wax into the tissues. Xylene will be replaced by paraffin, and putting paraffin into the tissues will make the tissues hard, and we will be able to section them or to cut them into small slices. Let's learn about fixation a little bit more. As I said, cells and tissues tend to get degraded by enzymatic action or by bacterial action once they are taken out of the body. Fixatives stabilize the cellular material and prevent autodigestion of tissues. These are the commonly used fixatives: formaldehyde, glutaraldehyde, alcohol, and acetone. In our lab, we use formaldehyde. Proper infiltration of the fixative into the tissues is necessary. Therefore, if you're taking a small piece of tissue, you have to take a container which is large enough to hold that piece of tissue and fill that with formalin enough so that the whole of the tissue block will be covered by this fixative. If you put a very large specimen into a small container and do not fill it properly, then fixation won't occur and you will get degraded or destroyed tissues. So if you want your final product to be good, you need to be careful when putting a tissue into the fixative. Now these fixed tissues are usually brought from the operating theater to the histological lab. And the fixed tissues we have to remove the water in these tissues and replace it with paraffin. So for this, as I said earlier, you have to dehydrate the tissue by passing them through a series of alcohol solution. If you just take the water out of the tissues, then the cells will shrink and you will not be able to see its structure properly. So you have to take the water out slowly by using alcohol solutions and replacing the water by alcohol. After doing that, we need to dissolve the alcohol using something called xylene. Then xylene gets replaced by paraffin wax. Now let's look at how dehydration is done. Here we use a gradient of alcohol. Initially, the tissue is filled with water. And then we have to reduce the water and increase the alcohol content. Initially, we start with 70% alcohol and slowly increase up to 100%. So the tissue is placed periodically into different solutions of alcohol. And finally, we say we bring it to 100% alcohol. So from water, we bring to 100% alcohol. Firstly, the tissue which has water inside it is placed in 70% alcohol, and then a series of alcohol solutions, and finally 100% alcohol, which tells us that the water inside the tissues have been replaced by 100% alcohol. Then we use what is called a clearing agent to clear the alcohol used, and the chemical we use here is called xylene. Xylene is a bit of a dangerous chemical and you have to use it in a proper lab under what is called a fume hood. Okay, so what this does is to remove the alcohol. So we put the tissue which has 100% alcohol inside it into a solution of xylene. We replace the xylene again until all the alcohol is replaced by xylene. Alcohol reduced, xylene increased. Then we can evaporate the xylene by heating it in up to 58 or 60 degrees Celsius. By doing this, we allow liquefied paraffin to infiltrate into the cell. So xylene is replaced by paraffin. Paraffin solidifies as it cools, and that makes the tissue a hard structure. So from a soft watery structure, now you have come to a hard structure which is easier to slice. So what we do is, in the xylene, we put this tissue into a wax bath and heated wax bath. So the heat will evaporate the xylene and the liquefied or the liquid paraffin will go into the tissues and replace the xylene. So finally, the tissues are filled with paraffin wax instead of water. Now, this long process was initially done by hand. You had a timer and you kept putting the tissues into different solutions. But we have what is called a tissue processor. You can see these containers here, which will be filled with the different solutions. Initially alcohol, then xylene, and then paraffin wax. So this is the paraffin wax chamber. This is the alcohol chamber. Like that, all this will be filled with chemicals and the tissue blocks will be inserted into this processor. So automatically, these tissues will go through each of these chemicals and it will take up to 12 hours, 12 to 15 hours for this process to occur. So you can put this machine on and the lab can be closed and people can go away, and when they come back, the tissues will be processed. Otherwise, it will be a very long drawn out process if you're trying to do it manually. So because we have a tissue processor, we can do this easily. So when the tissue is processed, you get a tiny block of tissue, which is now hardened. Then what you do is you put it into what is called a tissue cassette. Okay, to do this, you need a small container like this. And into this container, into this small hole, you put the tissue and then you fill the rest of it with liquid paraffin. You heat the paraffin, make it liquid and pour it into this, right? So when you pour it into this and then let it cool, it will set like this, like ice cube will set, right? So the wax will set and you have a block of wax, right? So the tissue is now embedded in a block of paraffin wax. Okay, and inside the tissues also you get paraffin wax. So the whole thing now is able to, it is quite large now, right? So if it was only this, it will be quite small, if it only the tissue. By embedding it in another block of paraffin wax, we make it larger so that it becomes easier for us to slice it. So this slicing, we call it sectioning, and we use a special knife to section this embedded tissue into very thin slices, very thin sections. So the technical term is sectioning. So how do you section? You use what is called the microtome. The microtome is used to cut the embedded tissue into thin sections, 1 to 10 micrometers. Usually we aim for about 2 micrometers, which is the easiest thickness to get. So when you take if you can take a section that is so thin that you get only one layer of cells, then light can pass easily through it and you can see all the structures. If it becomes too thick, then there will be several layers of cells and the structures will fall on top of each other and it will be difficult to see. So the microtome is there, and this is the handle of the microtome, and this is the knife of the microtome. So this, when you move the handle, the knife will move up and down. And here, this is the wax block which you have fixed to this area. And when this moves, thin slices will be cut and they will fall onto this area, right? So the video has been uploaded from YouTube. You can watch that and see how it is done. So here it is a close-up picture. This is the wax block which is fixed. And when you cut, you get thin slices of this. That is the tissue inside. This is the wax which is surrounding it. And this sort of like a chain, you will get this, right? So each of these can be separated and carefully placed on clean glass slides. Then what we do is, we take these thin sections, place them on glass slides, and then we float them on a warm water bath, hot water bath. So because of the temperature, the wax will melt a little and stick to the glass slide. So when that is done, you will take it and put it on a humidifier or a rack for it to dry. So when it is dry, the tissue is firmly adherent to the glass slide. Then you can put either a medium like a drop of water or saline and cover it with a cover slip, or you can make a permanent slide by putting a fixative and then covering it with a cover slip and actually sticking the cover slip onto the glass slide so that you will get a permanent histological slide. Now, this is actually a special machine called a cryostat sectioning machine, which you find in theaters. In Peradeniya Hospital, you find this. So this is actually used to take frozen sections quickly. So during a surgery, if you want to know whether some growth or a tumor is malignant or not, you can quickly take a section. You can quickly take a small part and cryostat it, that is, freeze that thing. So this whole process of making it solid by using paraffin and all that is done very quickly with this cryostat machine, where it freezes the tissue and makes it really firm so that you can cut it and look at it through the microscope, right? So this is done in emergency surgeries to check whether a tumor is cancerous mostly, right? Of course, you can use this for regular sectioning as well. Then, of course, the sections tissue can be looked at through the slide, but best thing would be to put color into it so that you can visualize more details. So we initially dye the tissues. We talked about this quite a lot. So you have all sorts of different dyes to color different structures, right? But the commonest dye is the hematoxylin and eosin, what we call HNE. So using these two chemicals, you will dye or you will color the slides so that you can see more details. Okay, so before you can actually stain these tissues, you have to rehydrate them. That is, you need to bring water because these two dyes are water-based. So the tissue sections have to be rehydrated before staining. In order to do that, we have to initially remove the paraffin by using xylene. So xylene will dissolve the paraffin. Then after that, we have to remove the xylene by using a series of alcohol and finally finish with water. So we are bringing the tissue back to water from paraffin. So once you do that, the tissue will absorb the stain. If you don't do this, because of the paraffin, the stain won't fix, right? So you have to remove the paraffin wax and bring the cells back to water in order to stain them. So this is the staining process. Again, this can be done manually or by a processor. In our lab, we do this manually, okay? So here you have different, again, you use xylene to dissolve the wax, then you use alcohol, and then finally up to water, and then you use eosine and hematoxylin baths, right? So this is eosine, this is hematoxylin, this is eosine, right? So to place, these are the these are the slides which are placed in this rack, and you can dip the rack into these staining baskets, right? So you stain them and you keep them to a regular time, and then you wash with water, and finally you have slides which are stained with eosine and hematoxylin and eosine, right? So this process is also there in the video. So finally, you will get slides like this, right? So this is a box of slides which have different tissues prepared and stained as the way that I have mentioned. Okay, so we studied this slide previously also. So hematoxylin and eosine. Hematoxylin is a basic dye which stains acidic structures purple. So it will stain the nucleic acid of the nucleus purple. Eosine is an acidic dye which stains basic structures pink. So it will stain basic cytoplasm in pink and the basic collagen in pink. Organelles also in pink, right? So here you see a slide which shows you nice pink coloring of the cytoplasm and nice purple coloring of the nuclei. This slide again I showed you. So this is a structure stained with hematoxylin. This is a structure stained with eosine.